just wanted to first thank everyone for being here today. My name is Cheyenne Sharif, and I'm the Associate Dean Research and Graduate Studies for the Ontario Veterinary College. And it is indeed my honor and pleasure to present to you this year's Chappel Memorial Lecture. I'm just going to give you a little bit of an overview of the Chappel Lecture. And we are very fortunate to have Dr. Chappel and Mrs. Chappel here today in attendance. And then I'll be presenting to you this year's Chappel lecturer, Dr. Laszlo Radvanyi. So first and foremost, the Chappel Memorial Lecture was established in 1988 through an endowment by Dr. and Mrs. Clifford I. Chappel in memory of Dr. Chappel's father, Herbert James Chappel. The objective of the series is to bring each year to the University of Guelph campus a scientist of international repute to address interested members of the student body and faculty. And through such an encounter and through further informal discussions, the university hopes to provide its members with an opportunity to hear people on the forefront in their own research sphere, who will not only summarize their current work, but trace its development and progression over the years. The Chappel Memorial Lecture is truly intended to be not so much a delineation of a field of study, but as a source of inspiration for those who are engaged in or considering graduate studies in biomedical research. Interested members of the public are also warmly invited to attend. So it is indeed with my great pleasure and privilege to introduce Dr. Laszlo Radvanyi. Dr. Radvanyi is the President and Scientific Director of the Ontario Institute of Cancer Research, or OICR, and he brings over 30 years of oncology research in academia and leadership experience in international pharmaceutical and biotech industries. He's also a professor in the Department of Immunology at the University of Toronto, where he performs research on the interaction of the immune system with tumor cells during the initial stages of cancer development to find targets for early intervention. Dr. Radwani joined OICR from EMD Serono, which is part of Merck, where he was Senior Vice President Global Head of the Immuno-Oncology Translational Innovation Platform and Senior Scientific Advisor for Immunology and Immunotherapy. And prior to that, Dr. Radvanyi was a professor in the Department of Melanoma Medical Oncology at the University of Texas MD Anderson Cancer Center in Houston for 10 years, where he ran an integrated clinical and basic cancer immunotherapy research program focusing on adoptive cell therapy. Dr. Radvanyi was instrumental in launching a pioneering company commercializing cell therapies for melanoma and other types of cancers. Dr. Radvanyi, with no doubt, is a sought-after expert in cancer biology and immunotherapy and a thought leader in the field of oncology research strategy. And I'm also delighted to tell you that he's been one of the biggest supporters and proponents of translational research and trying to marry veterinary medicine with human medicine in order to not only to find effective and efficacious methodologies and therapies for human cancer, but also to find similar types of therapies for companion animals and other types of animals. So without further ado, I'm just going to give the forum to Dr. Radvanyi to deliver this year's Chappel lecture. And then subsequent to that, I will come on board and provide some opportunity for our audience to ask questions and interact with Dr. Radvanyi. So without further ado, it's all yours, Dr. Radvanyi.
Thank you very much, Cheyenne. And again, I'd like to thank the Dr. Clifford Chappel and his wife and the Chappel family for supporting this lecture. I think it's really exciting, and I'm honored and excited to be joining you today. I wish it was in 3 dimensions or in person, but hopefully maybe next year I will come and listen to the next speaker and also maybe give other lectures on some of the other research I'm doing. So again, thank you very much. And I'm going to share my screen and go from there. And I presume everybody sees this. Somebody can frantically wave on their screen if they don't see it. So today I'm going to take you through a really interesting and exciting journey into what is called the dark matter of the human genome, which was elusive for many, many years due to a number of reasons. And this dark matter of the human genome is now actually emerging to be critically important in not only cancer initiation and progression, but also in how the immune system responds against cancer. And that has tremendous implications for immunotherapies, which really are the new and exciting approaches to treat cancer. And so today I'm going to talk to you about modulation of immune responses in cancer by retroelements and endogenous retroviruses, and which we believe are really exciting new targets for immunotherapy. So I'm going to give you sort of an overview of what retroelements are, endogenous retroviruses, why they're important, how they play a role in cancer, and then go into some of the past work I did when I was at MD Anderson Cancer Center with a number of colleagues, as well as the current research through a collaborative international network that I work with that's analyzing retroelements and their effects in retroviruses in various types of cancer. And I'll be showing you some really exciting recent unpublished data, which I thought would be really exciting for you to see. So these are just my disclosures. I do some consulting and SAB activities, etc. So essentially what we're talking about here is this thing called non-coding transposable genomic repeat elements in the human genome. We actually know that 50 to 70% of the human genome up until recently was considered, and I wrote in quotations there, junk. And as I'll show you in the next slide, this was unfortunate because there was a big paradigm for many decades based on some initial observations and publications by leading genomicists that the majority of our genome is simply junk, and that those small percentage of protein coding genes, which is about 1.5% as shown, together with the introns, which are 26%, that control gene expression, that's where all the action happens, nothing else. The rest of it is just fluff. But as you know, this has completely been turned on its head in the last a few years. And it turns out that human endogenous retroviruses and other long terminal repeat retroelements make up a large percentage of the genome, including things like long interspersed nuclear elements, short interspersed nuclear elements, human satellites, and as I mentioned, human endogenous retroviruses and their associated long terminal repeats. And I'll talk to you about the different mechanisms that these components use in regulating our genome. But you can see in this pie chart that the vast majority of our genome is in fact formed in these lines, signs, other retroelement HERVs, and DNA transposons. And only roughly 30% of our genome is actually what we call the coding genome, which produces the proteins that we all know about. So up until recently, these non-coding elements, as I mentioned, were the dark matter was originally thought of as junk DNA. And this notion emerged out of a landmark paper that was published by leading genomic researcher Susumi Ono in 1972, who basically promulgated the dogma or the idea that the vast majority of the human genome is junk, and that we cannot withstand larger sequences of coding elements, that the human or the mammalian species can only accommodate a certain amount of coding sequences, and that the rest of the genome is just simply hanging around as structural elements. And so this dogma of junk DNA really caused decades of lack of research and actually even ridicule of scientists who are trying to study it and try to say, well, no, wait a minute, this must have some sort of a role, because why, in fact, is it such a large part of our human genome? It just doesn't make any sense. Like, why are we wasting all the energy? If you look at all the ATP, the enzymatic activity during cell division that's required to replicate all this stuff during cell division, why in the world are our cells doing this? So I guess the as the field evolved, and now we're in the 21st century, 2022, I guess the lesson here, and one of the lessons for some of the young scientists here, is that facts don't kill theories. Only theories that exceed obsolete dogma can kill old theories. And I think that's really was really the story here of retroelements and endogenous retroviruses and their role in the human genome and in cancer and in other diseases, such as neurological diseases and metabolic diseases, where they also play a role. And as we know, these elements play a role in pregnancy as well, as I'll mention. So there's two major retroelements in the human genome, although there's others as well. One of them is these long interspersed nuclear elements, or LINE, or L1 as we call them. And in fact, these line elements actually code for open reading frames, which are called ORF1 and ORF2. And they actually represent 17 to 20% of the human genome. They're enriched in pericentromeric regions. And we have about 500,000 copies that we've accumulated during evolution. But these are mostly inactive and silenced. But about 150, what we call hot L1 copies, are actually active and transcribed. And as shown in the structural diagram of L1, of line 1, which shows the two open reading frames, they actually encode, especially open reading frame 2, encodes endonuclease, retrotransposase, and these other domains that actually allow these to replicate and then induce transposition of these sequences in different parts of the genome. So these line elements are actually able to replicate themselves and transpose themselves in various parts of the genome. And as you can imagine, this sets up a scenario where in pathophysiological situations of DNA damage, such as cancer, that these can cause havoc in our genome and can actually lead to genetic instability and mutational processes. So this is actually a very important component. I'm not going to go through this entire slide, but line 1 actually, and its retrotransposition, actually plays a very important role, or positive role, during embryonic development. And we still don't understand, but during the 2-cell to 4-cell stage, L1 is actually rapidly turned off and then rapidly turned off during the implantation of the embryo. And we still don't understand, but it may play a role in chromatin accessibility and ribosomal RNA expression. But then it gets inhibited, and it's actually highly regulated in germ cells during embryonic development. And for example, things like the Pee-wee Argonaute protein family, which actually degrades L1 transcripts. In somatic cells, when we grow up, line 1 is usually shut off by a number of genetic mechanisms, largely epigenetic mechanisms, such as hypermethylation at the L1 promoter loci by a number of different mechanisms, such as DNA methyltransferases, PRMT5, etc. And there's also a transcriptional repression by RUNX3 and other repressors. And in fact, what happens in cancer due to DNA damage and epigenetic modulation is that these transcriptional repressors that are usually hypermethylated get hypomethylated. And so the repressors that repress L1 then get shut off, and L1 gets expressed in cancer. And so L1, which is de-repressed due to DNA damage and epigenetic dysregulation, causes is induced, and there are transcripts and proteins actually detected in many types of cancers I'll show you. And there's hypomethylation to L1 loci, again, hypermethylation of L1 transcription repressors. And a lot of this is driven by things like oxidative stress and DNA damage. There are also pioneer factors that regulate chromatin structures, such as FOXA1, which is highly upregulated, for example, in breast cancer and endometrial cancer, which activate and induce expression of these retroelement L1 regions. And also, what's also very interesting, and as I'll mention later, is that a lot of these retroelements and human endogenous retroviral gene sequences are actually enriched in pericentromeric regions, which actually are the target of aberrant transcription factors such as the ETS family that gets overexpressed in many types of cancer. So it's sort of like a feed forward situation too. When you get expression of these retroelements such as line, they then get induced. They further cause DNA damage due to retrotransposition and disruption of intronic exonic sequences. They can cause stop codons to be changed, start codons to be changed. They will then alter RNA transcripts and splice sites, etc., and could lead to oncogene activation and tumor suppressor gene inactivation. This is just some, showing you how highly these L1 ORF1-2 protein is expressed, can be expressed. This is, for example, in bladder cancer on the left in comparison to normal bladder, which doesn't express these. You can see the dark immunohistochemistry staining. And across head and neck cancer, for example, there are a number of samples here showing ORF1 and ORF2 expression. So, one could then imagine that these can actually be perhaps tumor-associated antigens. And in fact, people are looking into this, as I'll mention later, that these are now emerging to be really exciting new targets for tumor immunotherapy through, for example, active vaccination against these epitopes arising from these ORF1-2 proteins. Now I'll just talk to you a little bit about human endogenous retroviruses, because this is sort of where a lot of my research has been done, which I'll be presenting to you. So human endogenous retroviruses are really ancient mouse memory tumor virus-like beta retroviruses that integrated into primates millions of years ago, about 2 to 55 million years ago. And as human evolution occurred from primates, and various Homo species evolved, there were cycles, boom and bust cycles of various retroviral infections in humans. And it then turns out that due to these multiple incidences over evolution of these retroviral infections and insertions into the genome, we now are in a situation where 8.3% of our human genome actually comprise of these ancient remnants of these ancient retroviral infections, which we call HERVs for short. And so our multiple HERV types that have come into humans over these millions of years and hundreds of thousands of years, including HERV-K, HERV-E, HERV-W. And in fact, what was interesting is that HERV-W encodes a protein, an envelope protein called syncytin 1 and 2. They're actually important for entropoblast fusion during placenta formation and normal pregnancy. And as I'll show you, these envelope proteins have immunosuppressive domains that are very important early during implantation and placental formation to prevent the mother from rejecting the fetus. So this is actually, so again, very interesting role of these HERVs in humans. The majority of HERV proviruses now have been inactivated over millions of years, hundreds of thousands of years, due to frameshift mutations, truncations, etc. But a more recent entry into humans is what is called HERV-K, and that is the most recent retroviral infection during evolution that affected humans about 100 to 200,000 years ago. And it turns out that because it's more recent, it had less time during evolution to get disrupted. And we have about now 30 to 50 copies of the HERV-K genome in us. And out of that, we have 10 subgroups, which we call HML1 to HML10, which is human MMTV-like. That's what the HML stands for. With HML2 especially existing as full-length active open reading frames yielding protein products, and what we call non-infectious pseudovirions due to the envelope protein that's encoded in the provirus. So HERV-K is actually now been very active, is beginning to be very actively studied by the cancer community, as well as communities studying other disease types, such as neurodegenerative disease, and for its ectopic induction during various pathophysiological conditions and what effects this may have. And so, and tumor immunologists and cancer researchers really ignored this area for many years. And when I was at MD Anderson Cancer Center, I happened to start a collaboration with a really dear friend of mine, Dr. Feng-Wang Yohanning, who's now in California. And we actually did a number of, quite a lot of work on the expression of these HERV-K in various types of cancers, and also how, because HERVs are not expressed in normal somatic cells, they get shut off, and then they get ectopically expressed in cancer, that they actually represent a whole new class of neoantigens for cancer immunotherapy. So this is the structure of HML2, the type 1, type 2 proviral structures. It actually is a full-length insertion. It has multiple insertion sites with a long terminal repeat, a GAG, POL, and LTR. And if you guys have been students of retrovirology, you'll probably tell yourself, wow, this looks very similar to HIV. And in fact, this is exactly the similarity. This is, with HIV and other similar type of viruses. I'm not going to go through the entire details of this slide, but suffice to say that there are two types of HERV-K, HERV-K1 and 2, and they differ based on a deletion of a 292 base pair fragment in type 1 that then takes away a splice donor site, which then is present in the type 2 HERV-K. And the depending on what splice donor sites can be used when active transcription is occurring off of these elements, you can actually get 2 additional transcripts in addition to the full length NP9 and REC transcript, as I showed you, based on the donor and acceptor sites. So one of the key genes that's expressed in human cancer and has been really well studied is what is called the ENV gene, which is the envelope gene. And this is a very interesting protein. It's actually expressed can be expressed at full length, and it actually has a signal peptide. It has a surface subunit, and it has a furin cleavage site that then that is cleaved, but then the molecule comes together using a covalent linkage on the cell surface. And this is a diagram, I think, of how this molecule is putatively thought to look like, where it actually forms trimers on the surface of cells that produce them when the ENV protein is induced. And it has this SU domain, and many of the antibodies that are made against the envelope protein of HML2 actually target this domain. There are some groups that are trying to actually target antibodies against the immunosuppressive domain in order to inhibit the proposed immunosuppressive functions of this molecule. But this is how it looks like. It's a transmembrane protein, but it also forms sort of budding vesicles, or sort of these pseudovirions on the surface. And as shown here, this is a picture from one of the publications that have studied this. It actually forms these envelopes, these non-infectious kind of pseudovirions formed by these envelope proteins. And these can actually then bind to other neighboring cells, be taken up by other neighboring cells, and can also, when they build up at high levels, can actually induce cell-to-cell fusion. And this is actually one of the, this is how syncytins, for example, from HERV-W, operate during trophoblast fusion early during pregnancy. So HERV-K sequences are usually inactivated or silenced in normal adult cells, as I said, because of disruptions in stop codons, etc., frame ships, etc. But because of DNA damage in cancer through chromosome breaks, frame ships, mutations, these sequences that usually stop HERV-K gene expression and ENV expression are removed. And then these transcriptional repressors and other stop codons and stuff can be removed, and then the HERV-K gets re-expressed in cancer. And as I said, they can be secreted in these budding structures, especially envelope protein can be and taken up by other cells. So they're not viruses, they're not infectious viruses. They don't have all the bells and whistles of viruses, but these sort of budding kind of pseudo-virion structures can be taken up by neighboring cells. And in fact, maybe even be part of larger extracellular vesicles too as well, that then can be taken up by other neighboring cells, not only locally in a tumor, but also systemically across the body of a patient. So Dr. Feng Wang and I started a collaboration at MD Anderson when I was a professor there from 2004 to 2014, and we got very interested in retroviruses. Back then, not many people were interested in them. They thought these were just oddballs here in cancer and don't represent anything important. And so we were kind of, stuck our neck out with a couple of other researchers in the area and started looking into this and how HERV-K RNA and protein may be expressed in different types of cancer. And I'm showing you just some snippets of some data we generated with Feng Wang, Johanning, showing at the transcript level, for example, in multiple breast cancer cell lines versus normal ductal epithelial cell lines, the expression of HERV-K envelope here, as here is an actin control. And also what's very interesting about human endogenous retroviruses and line 1 elements, which I probably neglected to mention earlier, is that they're induced, can be induced both in the prostate and the breast by steroid hormones. In fact, that's one of the interesting quirks of these. And we still don't understand the exact mechanisms, but steroid hormones, both androgens and estrogens, actually strongly induce the transcription of line 1 in many cases and HERV-K envelope in many situations. And this is actually shown in this breast cancer cell line here, plus or minus hormone here. You can see the induction versus the control. And we generated an antibody against the SU subunit, which is the surface, sort of the exposed subunit that comes, that's expressed from HERV-K called 686 or 586, I should say. This is just some snippets again of immunohistochemistry data we published showing that versus normal ducts that don't express, you see this expression of HERV-K envelope coming up in cancer. And this is just some more data on different invasive ductal carcinoma samples. So it turns out that you versus no expression in normal, and of course as you go from hyperplasia towards invasive cancer, you get a huge induction here of strong impression of HERV-K. This is just sort of amalgamation of a bunch of immunohistochemistry data from over 100 samples that we did. And this is another example in metastatic melanoma. You can see here controls here. In this case, this is red staining, red dye staining, showing quite a lot of expression of HERV-K envelope you can get. And we're also interested in how specific it is, and this is just a huge panel of normal tissues here. You can see here completely negative for HERV-K. So this actually represents a highly tumor specific protein that's induced in many cancers and very tumor specific. And as one would think, a good candidate for a tumor antigen for immunotherapy. This is again just a survey of a whole bunch of different types of cancer from prostate, showing in normal tissue no expression at all, and then high expression. For example, 100% we saw in 12 colonic adenocarcinoma samples, 92% in breast cancer, 90% in lung, and high percentage in melanoma and in prostate cancer as well. So these are just, again, some of the staining that we did for HERV-K in various tissues. So, okay, so HERV-K is expressed. What does it do and why is it important and how can we target it? So a number of researchers now have gotten very excited and involved now. And there's a community growing now studying retroelements and endogenous retroviral re-expression. And I guess so far, this slide is sort of shows you the culmination kind of what we know mechanistically of what HERV-K can do and how it can disrupt normal pathophysiology and its role in cancer. So, as I mentioned, the envelope protein has an immunosuppressive domain that actually has been shown to, when it interacts with antigen-presenting cells, to induce the generation of tolerizing dendritic cells, which then inhibit T cells. We don't know, again, a lot of how it's being done, but there's a number of studies that have been published to show that. And this is through the interaction of this HERV-K immunosuppressive or ISD domain, but it's still an unknown mechanism of action. Of course, the long terminal repeats and the multiple copies that can be produced from the long terminal repeats can go and get produced and then get inserted into other parts of the genome in introns, exons, and then these are actually full promoter elements that can actually then ectopically induce the transcription of oncogenes or disrupt the expression of tumor suppressor genes. So as you know, there's methylation defects in cancer, and these methylation defects then induce the expression of these HERVs, especially hypomethylation at the HERV loci, which occurs in many cancers. And there's also a link between envelope, but I'll show you some of the work we did, and the induction of oncogenes such as c-MYC and RAS, and the activation of the RAS-MAP kinase pathway. And as I mentioned, the hormones such as androgens and estrogens actually induce the expression of HER2, especially the REC and MP9. And these products has actually been associated with oncogene activation and immune suppression. And so these are, again, some of the mechanisms here through insertions, inversions, deletions, by ectopic insertions and transpositions of the LTR from HERV-K. But these other HML2-derived oncogenic proteins, as MP9 and REC, have been shown now in a number of studies to activate MAP kinase signaling, induce c-MYC, turn on beta-catenin, which then are all promoting cancer cell growth. And of course, the LTR activation of oncogenes due to these transpositions. And I mentioned the immunosuppressive domain again. IL-10 is a key cytokine that actually has been shown to be induced. So we actually, together with Feng Wang, did a number of studies. This is an example of one of our publications when I was at MD Anderson. And we looked at HERV-K viral envelope RNA and protein in pancreatic cancer. And it turns out that it's highly expressed in pancreatic cancer. And we and others have found that, and this is just showing you a number of pancreatic cancer cell lines here, the protein expression. And we've also done immunofluorescence staining of a number of different pancreatic cancer cell lines. And you can see the high level expression. And very interestingly, in many of the tumor cell lines, this is PANC1, PANC2, this actually can be found on the cell surface to a certain extent. And as I said, the envelope protein it's a transmembrane protein with a signal sequence, and it can actually then butt off the surface as these sort of vesicle-like pseudovirions, non-infectious particles. And I think this is what this sort of staining is really showing. And actually, as I'll show you later, this actually offers us an opportunity to target HERV-K using, immune-based methods, such as antibodies, for example. And I'll show you a little bit of data on that. And so again, these are just some more immunohistochemistry showing in pancreatic cancer, some more data showing, no expression of HERV-K envelope in matched uninvolved tissue. And you can see here, poorly differentiated, moderately differentiated PDAC, pancreatic adenocarcinoma, the expression of HERV-K. And as I said, it detected on the surface. We went on and we said, well, what does HERV-K do if we knock out the envelope? And so we made a number of shRNAs in lentiviruses to knock out or knock down, I should say, HERV-K envelope. We couldn't knock it down completely. And we did a couple of experiments by transplanting human pancreatic cancer cell lines with either wild type or with control shRNA or HERV-K-env shRNA. And as you can see, interestingly, we found that by knocking out HERV-K envelope, knocking down, that is, we actually can inhibit tumor growth. And this is just showing here in two xenograft models here, the tumor sizes that we get compared to the wild type, or non, control shRNA. Again, showing this. And it turns out that when we looked at more closely at these links between HERV-K envelope and oncogenic signaling pathways that turn on MAP kinase and other pro-proliferative and anti-apoptotic pathways, it turns out that by knocking out HERV-K on, we're actually inhibit ERK activation, RSK activation too as well, and in these different pancreatic cancer cell lines. And we looked at the levels of expression by quantitating this data. You can see here that HERV is being knocked down by the shRNA. RAS is being affected, but as we found that actually c-MYC, phospho-AKT, and some of these MAP kinase activators are also inhibited in their activity. So although it's not a 0-1 in terms of we're completely knocking out activation of these pathways that induce proliferation and survival of cancer cells, it seems that the envelope proteins having some significant contribution to oncogene activation. And again, we're still working on trying to understand, what that is. So the next question, as I hinted, are L1 and HERV-K proteins immunogenic, and can they be targeted for immunotherapy? Obvious question, since they seem to be very specifically expressed in cancer cells. And the question is, it seems so. This is not our data, but this is just to touch upon line 1, retroelement expression. There's been some recent exciting data. This is Solovyov et al. Nice paper in Cell Reports. They did a TCGA analysis across 30 tumors that had total RNA sequencing. Not many of the TCGA samples had total RNA as opposed to poly-A RNA, because again, people are not interested in the non-coding region. So they didn't do total RNA, but they did do total RNA in a subset of tumors in the TCGA, which would then contain these retroelements and resulting transcripts. And so it does turn out that this group found that the higher L1 retroelement expression across these 30 cancers, which included samples from lung, breast, urothelial, and colon cancer, the higher L1 expression correlated with much better survival, overall survival in the TCGA database. Again, low numbers of samples because of the paucity of the data and the data generated. But again, this is something that I think we need to follow up on in many more samples. Again, high L1, better survival, and yeah. In this, yeah, I think this is and so the other interesting thing that they found was that in low, and this is L1 immune. They looked at the immune scores relative to L1 expression, and they found that there's a correlation in that, ironically, in the low L1 expressing tumors, there were found more CD8 cells than in the high L1 expressing tumors. And that correlated to survival. And one would say, well, wait a minute, that's contradictory to what I was just saying. And the explanation that they gave in this paper is that the L1 is inducing a strong CD8-mediated immune response and tumor infiltration, which is then killing off the cells that are expressing high levels of line 1. And so what remains is cells that express very low levels of L1. And so there's a sort of an immunoselection occurring in these tumors against high line 1 expressing tumor cells. So an interesting mechanism that, I think people should follow up on. So together with Feng Wang Yohanning and of course her team and my team, we did quite a lot of work. And these are just some snippets of some of the publications that came out from our labs when I was at MD Anderson. And we started looking at human endogenous retrovirus protein expression and whether we can induce immune responses and whether patients endogenously or de novo launch immune responses. I mean, if these are sort of foreign-like antigens that are not expressed normally in cancer, one would then argue that, they would be immunogenic and represent neoantigens. And so we did quite a lot of work across melanoma, breast cancer, ovarian cancer, and did some work on pancreatic cancer, as I showed you before. So the way we did these experiments is, this is a patient with cancer, is we got blood samples from these patients, and we would isolate blood samples from these patients. We would also isolate the tumor from tumor resections. We would get fresh tumor resections. And then we would mechanically and enzymatically dissipate these tumors into single cell suspensions. And then we then preserve these or grow these as cell lines to then use them as targets to determine if they are recognized by antigen-specific T cells. And so what we did with the PBMCs is that we generated dendritic cells using a typical procedure, using adherent monocytes and GM-CSF and IL-4, and then maturing them with the cytokine cocktail. And then we would then keep the other PBMCs as a source of T cells. And we would then use a number of RNA that express or not express different transcripts, in addition to, for example, the HERV-K envelope SU control proteins that we use, the HPV16E6 as a control protein, or KLH. And then we would then take these transduced dendritic cells, in vitro re-stimulate the PBMCs, the T cells, against these. And then about 7 days later, then we would test for the effector function of these T cells against breast cancer cell lines that were HLA matched, or samples of the tumor cells that we would isolate from the freshly, resected tumors. And so we looked at both, and then at the same time, from the serum of these blood samples, we looked at whether there are actually antibody responses that are induced against HERV-K, as well as in addition to the T cell responses. And this is actually a Western blot of sera from a number of breast cancer patients. This is our control. This is the antibody against the SU-ON protein that we generate, the monoclonal. And you can see here, this is like an example of just some of the breast cancer patients. You can see here the positive blotting. So these are IgGs in the serum of breast cancer patients. And then we went ahead and did a whole group of breast cancer patients. And you can see here the different dilutions of the serum versus normal donors, is that there's actually an induction of an appreciable serological response against HERV-K envelope. So there are antibodies against HERV-K envelope in these breast cancer patients, in the majority of breast cancer patients. And then after in vitro stimulation of these T cells, we did what are called interferon ELISpot, in which we then incubated the in vitro restimulated T cells with dendritic cells expressing either control antigens or the subunit antigen. You can see here, we can see, quite an intense interferon gamma response that can be seen in these example patients. And of course, we did these with a whole bunch of other patients. This is a normal donor here, where you don't see this antigen-specific response. And we did CTL assays against HERV-K envelope positive breast cancer cells that are HLA-A2 matched, such as MDA231. And you can see here, relative to, various controls, I mean, it's not the same in all patients, obviously. That's why, patients are not killing their breast cancers with CTL responses against HERV-K. But we saw here a heterogeneous yet appreciable CTL response. And this is just a snippet of some of the data. We have more data in the published paper here that we published in Cancer Research in 2008. And these are just, some of the things. We went ahead and did this in ovarian cancer too as well. This is an example, again, of serum dilution of patients showing recognition of HERV-K envelope. And this is just, again, at one of the dilutions to show the differences between benign, normal versus tumor-bearing patients. And again, this is an example of the same kind of system, stimulate T cells, and then look at their antigen-specific response using interferon ELISpot. You can see here an example of one patient versus a normal donor. You can see the induction of HERV-K interferon gamma response. And similarly, we showed that these ovarian cancer patients have T cells that can actually generate CTL that can kill tumor cells in an antigen-specific, HERV-K specific way. So again, very exciting, and it sets up the scenario of targeting HERV-K envelope in an immunotherapy. Now, as I mentioned earlier, HERV-K is also expressed on the surface of melanoma cells and other cells, and we went ahead and generated CAR T cells, and so we also showed in this paper in collaboration with Lawrence Cooper's lab at MD Anderson that you can actually generate CAR T cells that recognize HERV-K-ONV SU. And although it's not a clean win, this is just a single infusion of CAR T cells in this sort of xenograft model. You can see that you can actually get appreciable tumor control and inhibit metastases. The other interesting part is that murine ERVs are also expressed in murine tumors, such as CT26 and MC38. And these have actually been shown over the years to be sources of rejection antigens in many of these, such as B16 melanoma and some of these other colonic tumors. And so these HERVs and the immunogenicity of these ERVs also are found in murine systems as well. I'm going to skip that. So the other interesting thing about HERVs is that one of the exciting developments that has been shown with regards to HERVs and cancer therapy is that a new class of drugs called DNMT inhibitors, or DNA methyltransferase inhibitors, that control epigenetic modulation in cancer and sort of inhibit gene methylation so then the cancers express more genes, can differentiate and express more antigens. It's a new therapy. That this therapy has been found now to actually induce endogenous retroviral gene expression, which then actually induces a type 1 interferon response because of the double-stranded RNA, a surge of double-stranded RNA, producing type 1 interferons. And then these type 1 interferons then can actually affect tumor cells and immune cells and activate them through various pathways to increase immune recognition of cancer. And it was actually through a chance observation by Steve Balin, who actually is a world leader in DNA methylation, who did trials with DNA methylation inhibitors in lung cancer patients. And he paradoxically found that when he treated a group of patients that were previously treated with these inhibitors that then progressed, and he treated them with anti-PD-1, which is his checkpoint agent, that many of these patients actually responded to this immunotherapy. And this then opened up this whole area of investigation of how DNA methyltransferase inhibitors are now actually activating HERVs and then inducing these type 1 interferon innate immune responses. The question then becomes, if this happens, and the other question largely, if tumor cells are producing all these retroelements, HERVs in line 1, and in certain cases when they're treated with drugs, they actually induce high levels of transcription off these retroelements, and why aren't the tumor cells themselves inducing a type 1 interferon response? And as we know, as many of you know in tumor biology, tumors are relatively resistant to induction of type 1 interferons. It's well documented that they're defective in induction of type 1 interferon responses. And in fact, the lack of interferon response in tumor cells is associated with the sensitivity of things like oncolytic viruses. And also it's been shown that oncogene activation, things like KRAS and c-MYC, has been shown to block type 1 interferon responses. So the question is, are there other mechanisms of resistance in tumor cells to retroelement and HERV expression in terms of its induction of type 1 interferon, and why are the tumor cells themselves resistant to this? Why aren't they inducing type 1 interferon and becoming, being recognized by the immune system better? So one of the questions we started asking, because of our interest in extracellular vesicles and because we know tumor cells secrete copious amounts of these, is that can tumor cells actually package and secrete retroelements and HERV RNA in extracellular vesicles? And can that actually be a way of purging or getting rid of these retroelements out of the tumor cell to protect the tumor cell? But then indirectly, can that act as a systemic form of chaos in the immune system, because these extracellular vesicles can actually be taken up by other normal immune cells and stromal cells, and then these retroelements will be unleashed in these cells, and what effect may that have? And that's never been questioned or never been looked at. So we actually, and I'm not going to go through extracellular vesicles, I think many of you know, extracellular vesicles are not only biomarkers, but they're now shown to be active drivers of tumorigenic progression. They've been variously called microvesicles, exosomes, but I think the community now is accepting the term extracellular vesicles due to a certain amount of heterogeneity in their content and structure and size. But they can house DNA, RNA hybrids, microRNAs, mRNAs, proteins, lipids that can actually be taken up by other cells and affect signaling and other physiologic processes in these cells. And of course, we can do a whole bunch of multi-omic tools, proteomic, genomic, transcriptomic, to analyze what's in these isolated extracellular vesicles. And so they have a role in cancer. They're secreted by cancer cells in high amounts. And they actually can become a tumor marker even by measuring them in the blood for tumor burden. The more extracellular vesicles from the tumor, the higher the tumor burden. So now they're being looked at as early markers for recurrence and or the effects of therapy. They're loaded with proteins, lipids, and RNA, which when taken up by different normal cells, including immune cells, can disrupt normal functioning of immune cells, including inducing immunosuppression. And so part of our research goal was to study a number of tumor types, including Ewing sarcoma, which is a really highly untreatable sarcoma in kids, prostate, breast, and breast cancer, and to which we know induce extracellular vesicles, isolate these extracellular vesicles, and then study what effect they have on different types of cells that may take up these extracellular vesicles, and also do deep multi-omic analysis of what's in these extracellular vesicles, especially in terms of these retroelements and HERVs. And then in patients that have high tumor burden, especially patients with Ewing sarcoma and other cancers, we also isolated extracellular vesicles from the plasma and then did these same experiments. We did functional studies on the effects of ZEVs on target cells, and also looked at what sort of genomic elements are in these extracellular vesicles versus the parent tumor cells. I won't go into Ewing sarcoma. Ewing sarcoma is a highly aggressive cancer. We focused a lot on this area through an international collaboration. It's a pediatric childhood malignancy. 25% of diagnosed patients are metastatic at the time of diagnosis, and the survival is dismal. Less than 20% survival rate with patients with and with metastatic. The only chemotherapy is possible. You can see here this sarcoma usually goes in and around the bones, and you can see here multiple metastatic areas in these patients. Very dismal disease. Ewing sarcoma is actually what we call sort of an oncogene-addicted tumor that's induced by a chromosomal translocation between chromosome 22 and 11, between the EWSR gene and the FLY1 gene, that then induces this ectopic fusion protein called EWS-FLY1. And these FLY1, ERG, ETV, and E1AF and FEV1 are also targets for other translocations that constitutively activate. And these are S family transcription factors that actually are well known to drive cell proliferation, cell survival, and this is largely what drives the proliferation and the survival of these Ewing sarcoma cells. And these Ewing sarcoma cells are also known to be immunologically cold. They have what are called mostly a lot of macrophages, but they're so-called M2 macrophages, which are sort of suppressive macrophages. And these patients are characterized by having a lot of systemic inflammation, but at the same time, they're highly immunosuppressed and susceptible to infection, etc. I just wanted to point out, I'm not going to go through this whole slide, but these fusions that drive aberrant ETS protein transcription factor expression is not only in Ewing sarcoma, but also occurs in a significant percentage of prostate cancer and acute myeloid leukemia. And I'll talk a little bit of prostate cancer because that's a big, problem, still an unmet medical need in our society, with many older patients now getting prostate cancer. And this results from the fusion of the TMPRSS2 gene with the ERG gene, which actually activates this ETS factor translocation and activation. So suffice to say that many cancers now have these translocation. And what these translocations do, especially aberrant ETS activation, is they trigger a lot of transcription off of a number of chromosomes at pericentromeric regions. And it turns out that these pericentromeric regions are highly enriched for human endogenous retroviral gene copies, as well as line and SINE, human satellite DNA, and other retroelements. And so we started looking at these, and this is just a schematic of how we isolate extracellular vesicles. And this is just showing that we're isolating purified extracellular vesicles with the typical EV markers such as annexin, syntonin, and lack of common contaminants from the cytosol here. And so we're looking at pretty pure extracellular vesicles from the cell lines and the plasma that we are studying. And one of the first things we realized is that in metastatic and localized Ewing sarcomas and their extracellular vesicles versus extracellular vesicles isolated from healthy individuals from their plasma, and these are plasma samples, is this huge abundance of repeat DNA sequences and so RNA sequences, transcripts, versus a typical low level of these transcripts, of these retroelement transcripts, including line, SINE, and HERVs in healthy donors. And so, and another interesting point is that, getting to the question I was asking, is that in the tumor cells themselves, the abundance of these repeat retroelements are much lower than in the secreted extracellular vesicles, which suggests that somehow the cell is getting rid, perhaps, of these expressed lines and SINEs and HERVs through secretion, through these packaging and secretion through these extracellular vesicles. And so that's sort of a hypothesis that we're further following up on. Another important point that we found is that and these are again cells that we isolated directly from Ewing sarcoma patients. We isolated T cells and myeloid cells and B cells. And we then looked for the expression of HERV-K and human satellite RNAs, these retroelements. and we found in the myeloid cells, these are the CD33 positive, highly enriched for these retroelements, which were not found in the CD19 cells. Interestingly, we found retroelement expression in CD8 cells, especially activated CD8 cells. But this we found is not due to tumor extracellular vesicles infecting these cells. We went on and showed that, in fact, the extracellular vesicles from Ewing sarcoma and other cancers can infect myeloid cells, but not CD8 cells. And so we think that there's an endogenous activation of these retroelements in CD8 T cells, specifically when they get activated. And this is actually a novel finding we're following up on. This experiment simply is taking these vesicles from healthy donors or Ewing sarcoma patients, and we then treat various PBMC populations. And this experiment is showing that, especially the myeloid cells, this is HERV-K HSAT, are really the ones that are taking up these vesicles. And then what's interesting is that it's inducing these cells to actually then not only express, even show expression of these RNAs from these vesicles, but the cells themselves start expressing themselves HSATs and HERV-K. So so the actual EVs, what we're finding, actually induce the host cells that are taking up these EVs to then activate transcription off of these retroelements themselves within the cell, not just from the EVs. So what we're finding is it becoming an infectious cycle. We've done further experiments in which we've isolated EVs from the infected myeloid cells and then treated other fresh myeloid cells and showed that then it induces so there's feed-forward effect. And I'll skip this, but it just, in a xenograft model, I'll just briefly say that we also can see that even in the mouse stroma, in an immunosuppressed mouse model, a xenograft of Ewing sarcoma, we can actually see HSAT expression using in situ hybridization, both in immune cells and in the fibroblast. And what we find is that this is an experiment where we isolated EVs from a Ewing sarcoma cell line and treated fibroblasts, that these EVs actually induce a type 1 interferon response, innate immune response, through activation of CGAS, a RIG-I response, and also inducing, DNA damage checkpoints such as CHEK1 phosphorylation and CDC2 phosphorylation. And in fact, this is a stain for what is called gamma H2AX, which is a marker for double-stranded DNA breaks. And what we found is that when these stromal cells, such as MRC5 cells, take up these Ewing sarcoma EVs loaded with these retroelements, they actually cause DNA damage, probably because these RNAs we suspect, get reverse transcribed and then inserted into the DNA, causing a lot of DNA damage and havoc. And you can see here the immense H2AX staining. This is with etoposide, is a control drug that induces DNA damage, but you can see here the damage that's induced by these EVs given at different concentrations to these MRC5 cells. The EVs induce a pro-inflammatory response in both myeloid cells and fibroblasts. Again, same experiment where we treat EVs. These are MUTS3 myeloid cells and MRC5 fibroblasts. MUT cells are very nice sort of monocyte-like cell line that can be differentiated into macrophages or dendritic cells. And then we treat them over different lengths of time with isolated either Ewing sarcoma plasma EVs or with EVs from Ewing sarcoma and other cancer cell lines. And you can see here the induction of pro-inflammatory innate cytokines such as interferon beta, TNF, IL-35, IL-20, IL-8, etc., osteopontin, both in the MUTs and in the MRC5 cell line. So suffice to say that these EVs containing these retroelements are turning on quite a powerful pro-inflammatory innate immune response. And you can imagine in the patient, if this is occurring over and over again, where the tumors are growing, secreting EVs. EVs are being taken up by these normal cells throughout the body and in the tumor microenvironment. They're inducing a type 1 response. Then there's this inflammatory response. Then those cells that are infected may actually producing EVs themselves that then feed forward. So you can see how this is setting up this sort of smoldering response in sort of storm in these patients, this smoldering underlying inflammation in these patients that can be quite immunosuppressive. To sort of double back, and these are, I'm almost finished here. I'm going to get to the end pretty soon because I know that we're getting low on time, is that we can actually transfect these fusion genes like EWS-FLI1 into HeLa cells and actually show that we get the same effects as when we isolate EVs from the cell lines and from the plasma of these patients, where we're actually inducing these microsatellite retroelement RNA transcripts, and as shown here, and also inducing type 1 interferon and type 2 interferons in response to the transfection. HeLa cells don't normally express these retroelements, so they're a good control cell to use in this regard. Just to suffice to say that in prostate cancer, as I mentioned, has this fusion, this TMPRSS2 EGR fusion, which also is in which together with testosterone activation in prostate cancer induces, as shown here, a significant amount of retroelement expression lines, SINE and HERVs as well. And here in a gene expression analysis, we show that this induction with testosterone induces a type 1 interferon response, as well as a DNA damage response. And of course, when we knock down EGR, we also knock down EGR with siRNA. We actually can show that these retroelements are in response to androgens in these prostate cancer cell lines are actually knocked down. So there's then this link between androgens, the expression of this fusion, induction of type 1 interferon, and we've actually shown that in these prostate cancer cell lines, that these cell lines also secrete extracellular vesicles that then are loaded with these retroelements that can infect myeloid cells and fibroblasts as well. So I'm going to stop there, and I'm just going to say, well, where do we go from here? So we have pretty good evidence that retroelements and HERVs, I think, in cancer may drive an underlying smoldering systemic inflammatory state characterized by adaptive immune suppression. We published other papers with our collaborators to show that these EVs from Ewing Sarcoma patients, for example, can disrupt normal dendritic cell maturation and generate what are called sort of tolerizing dendritic cells that don't stimulate T cells properly. So I haven't had a chance to show you those data. So, and I've also shown you data to suggest that tumor cells may be sort of purging these retroelements, HERVs, HSATs in EVs as a critical mechanism to not only protect themselves, but then through secreting these elements, induce this chronic inflammatory dysfunctional state. And so what we're suggesting is that immunotherapies that are now in the clinic, many of them, that are sort of intended to activate and bolster the antigen-specific adaptive immune response or T cell responses through things like checkpoint blockade, vaccines, co-stimulation, may be failing to a large extent in many cancer patients due to this underlying smoldering inflammation, which then also drives T cell exhaustion, improper myeloid cell differentiation, etc. And this is maybe why, you know, a lot of patients have what are called these cold tumors and don't respond. And this actually suggests a completely new paradigm shift in immuno-oncology. in the way we treat patients. Because what we're then trying to do, given this situation, is that we have these patients that have this underlying smoldering innate inflammation that's inducing immune suppression. And then on top of that, we're trying to add immunomodulators that activate the adaptive immune response. And it's not working properly. So I think maybe a new paradigm shift in immunotherapy for cancer is that maybe patients should be pre-treated with a dose or with of anti-inflammatory drugs that reduce this innate smoldering inflammation. In other words, paradoxically, drugs that block type 1 interferons, drugs that block IL-1 beta, IL-6, and TNF to sort of lower this inflammatory state in patients, sort of reset the system, and then come in with vaccines, checkpoint blockade, and some of these other immune stimulants that we want to activate the adaptive immune response. And by this paradigm shift, although it's a challenge developing it in the clinic, but I think there are some people interested in this, we might be able to turn the tables in terms of inducing much, much better adaptive T cell responses against cancer by lowering this sort of chronic inflammatory state. And so I'm going to stop there, and I'm just going to just acknowledge my past collaborators at MD Anderson. And I really have some amazing people in my lab: Valentina Evdokimova, Peter Ruzanov, who's a bioinformatician, Valentina, their husband and wife team. She's the biologist that has done a lot of the work on the endogenous retroviruses and EVs in the cancer cells I showed you. And of course, I have a very talented technician and our collaborators. So I think I'll stop there because I went probably way over, and I apologize for that.
No, that was wonderful. And thank you very much, Dr. Radvanyi. It was just wonderful, and I'm sure that there are a lot of people wishing to give you a round of applause, and I see emojis popping up. So thanks so much for this wonderful talk. We are indeed over time. However, the good thing is that we've actually scheduled this webinar until 2 p.m. So for those of you who have to leave for whatever reason, for other commitments, please go ahead. We do have another 10-15 minutes or so that we can entertain questions. If Dr. Radvanyi, if you're available and willing, we are more than happy to keep this webinar open for the next 10-15 minutes or so. And from the audience, if there's some burning questions, please just raise your hand, and I think you can unmute yourself. John, Dr. Jonathan Lamar from Department of Biomedical Sciences. John, go ahead.
Hi, Laszlo. I'm just jumping in here because I got to run to another meeting, but I'm fascinated by your whole paradigm, and we don't often, my lab at least, doesn't think often about immunity with respect to those things like true classic innate immunity. We're, as I told you this morning at the meeting, we're really interested in the P-weak pathway. I wonder if you looked at the cells that were left behind. You said you can't drive it to zero when you activate an immune system against say a HERV protein expressing cell, for example. In the ones that are left behind, do they have some of these P-weak-like or other suppressive mechanisms? So you said it's like an immunologic selection that we're applying to these tumor cells. One of the things that I might argue that they're selecting for is a pathway that represses TE expression, or any of these TEs. So you've treated them with something that wipes out those that are expressing, say, line, open reading frame 1, or a HERV protein at their surface. Now what you're left with are those that have the ability to shut down those TEs. I'd be interested in your thoughts.
Yeah, no, so we haven't looked at that, and I think that data I show you with line 1 and that TCGA analysis paradoxically does suggest that hypothesis, where there's an immunoselection occurring, and that occurs with other antigens, neoantigens too, as well. With, and this is the paradigm or the paradox of the immune system, is that it's great, but it's also a fancy way of just selecting for more resistant cells that don't express the antigens that the immune system recognizes. And so, this is definitely going to become probably a resistance mechanism when we're trying to target these retroelements and HERV-K. And then we're going to have to look at the residual cells and carefully look at, what they're expressing, how they're expressing it, why they're expressing these retroelements, and perhaps how we can get them to re-express. And this is why combination therapies, such as with oxidative damage, sodium arsenate, for example, is known to induce retroelement expression, for example. So we need to then figure out pharmacogenomic means or pharmacogenomic screens, how we can re-induce retroelement expression in those residual cells and then make them immunogenic again. I think that could be a very interesting drug discovery paradigm.
Yeah, excellent. Thanks very much for your talk. Really enjoyed it.
Very good. Thank you, John. Dr. Alicia Villoria Petit from Biomedical Sciences.
Hi, I really enjoyed the presentation, particularly the part about the role that extracellular vesicles may be playing in this whole process. So I was wondering if you have looked at the role of those EVs containing those retroelements in metastasis, since metastasis is really what is important for patient survival.
Yeah, well, I mean, most of the patients, for example, the Ewing Sarcoma patients that we got samples from, I mean, invariably a large proportion of them are already metastatic. And, the samples of prostate cancer, I have to recall where we got some of we've done work with prostate cancer plasma too as well from patients, and a lot of those samples are from metastatic patients. And some of the tumor lines, as you know, many of them are from metastatic situations. So to answer your question, we haven't really clearly delineated early versus later stages of cancer and how the quality and quantity of these extracellular vesicles changes during different stages of cancer progression. I think that's going to be a very interesting thing to look at. But we do have some data, as I showed you, where there seems to be a higher level of EVs and retroelement containing EVs in more advanced stages of cancer. That could be simply because of tumor burden, higher turnover of the membrane, etc. It may be, an indirect effect. We're going to have to see.
That's very interesting. And my other question is about the mechanism by which these vesicles are released from the cell. Is it similar to mechanisms that have already been published? Like you mentioned something about budding of the membrane.
Yeah.
So is it a mechanism that could be suppressed somehow because we know how it works?
It's possible through antibodies. So this is one of the ways which maybe we could suppress it or sort of sequester these sort of enveloped circularized structures of ONV protein by binding them with antibodies. And maybe paradoxically, there are good and bad antibodies. And so some antibodies are neutralizing, some antibodies are not. And we haven't looked more deeply at the IgG isotypes in these patients that mount serological responses de novo against HERV-K envelope. It could very well be that, as you know, they could be antibodies that actually are bad because they're binding to HERV-K env extracellularly, and they're actually promoting the infection of additional myeloid cells with Fc receptors, as opposed to neutralizing. And that could actually be another mechanism by which HERV-K envelope causes immune disruption. But that's an avenue we and others I don't think have looked at, but something that should be looked at. So sort of antibody-dependent enhancement that we think actually happens in certain viral infections too as well.
Thanks.
Thank you, Alicia. Any further questions for Dr. Radvanyi? I know, Laszlo, at some point of time we had at our peak, we had 75 people attending the seminar, so it was well attended. I can tell you that much, and I just confirmed that they came from all over the country. So, like I said, one of the, I guess, benefits of the pandemic is that now we have further and more accessibility for our seminars. So I'm just going to go once.
Cheyenne, there is a question in the chat.
In the chat, okay.
Yes. So it says, how similar could HERV-K pseudovirus particles be to DI particles, defective interfering particles, And are they discriminative in their ability to infect host cells?
I'm not sure what you mean by defective interfering particles. So maybe I don't know if the person's here and can tell me what he means by defective interfering particles. But yeah, maybe.
Nitish, are you still there? He might have already left.
Yeah, maybe I can touch base with him later. But if I understand the question correctly, yeah, I'm not exactly sure. So I probably need to answer. I need to talk to him as to what he means by defective interfering particles.
Well, if you wouldn't mind, he's actually my research associate, so I can tap on his shoulder and let him know that he can send you the question.
Yeah, please
in your private email.
Let him send me the question directly. Yeah, by email.
Yeah,
Definitely
we'll do.
We'll do. Yeah.
We'll do. I don't really see any other questions in the chat. And there was a comment by Malcolm. Superb seminar, Laszlo. A real tour de force. Fascinating, thought-provoking, and inspiring.
Yeah, great. Yeah.
I thought you may not actually have access to the chat since you're an external guest. That's why I was reading it off the chat. Wonderful. Any further questions for Dr. Radvanyi? I'm just going to go once, twice. Oh, Alicia. So please, and maybe the last question is going to go to Alicia.
Sure.
Yeah, I didn't want to overtake all the questions, so that's why I just, went away for a while. But I wanted to ask about, so there are these therapies that induce immunogenic cell death.
Mm-hmm.
And these include even some chemotherapeutic drugs that we know, right? Had you look at those strategies that we know induce immunogenic cell death, and whether this is one way of inducing this type of expression of these retroelements?
Well, that's a very good question, and I was, tying into the question, the answer to one of the previous questions, is that this could be a new kind of drug discovery kind of avenue where we if these are indeed immunogenic, and if we can then do combination therapies where we can eliminate these retroelement HERV expressing cells because they're really powerful tumor-associated antigens, then if we can find ways of augmenting their expression and release, but then it's a two-edged sword, because then if we also are inducing a lot of exosomes and EVs that then cause immune disruption, then we got to be careful. But then immune responses, if we have an antigen-specific immune response, technically it should eliminate those defective immune cells too as well. But I mean, I do agree with you that if we can immunogenic cell death, whether immunogenic cell death has been tied to specific recognition of these retroelements and HERVs, I don't think that link has been made yet, and that hasn't been studied, and whether that's an important component in the downstream immune response tied in with immunogenic cell death. I think this is something that we need to look at, because the way the cell dies, the way it packages things in the dead, dying body of a cell through either apoptotic bodies or released antigens, etc., that is going to be very important in the way surrounding myeloid cells and dendritic cells pick up antigen and process and present the antigen. This is why things like necroptosis, for example, efferocytosis and things like that are now becoming of interest in the Gas-Merden pathways, because they seem to be inducing more of an immunogenic form of cell death, where traditionally our interest in apoptosis was actually kind of barking up the wrong tree, because in many cases apoptosis is actually tolerizing rather than immunogenic. But this is something definitely, I mean, there's a lot of things that this opens up for us to study.
Yeah, that's one of the reasons I was asking you about the mechanism of release of the particles. Because, apoptotic bodies are also vesicles, right? So, I was,
right.
Sometimes this, activation of retrovirus could cause some sort of like apoptotic response in some cases. So.
Correct. Yeah, we don't know. I mean, this is something we could look at, whether these retroelements are in apoptotic bodies, depending on the size. Generally, I don't hear, I mean, I may have missed it, but I don't see any reports on that. Definitely they're in these EVs, huge amounts in these EVs, which was really surprised us when we saw this. We were just floored. And then of course the infectious sort of infectious nature of these, which I think explains a lot, especially in late stages of cancer. As you know, in late stages of cancer, one of the major problems is, innate inflammatory cytokine-induced cachexia. And this actually, could largely be due to these EVs that are being produced in these situations.
That's interesting. Maybe it's important to investigate that with, it can be done with plasma, right? So, yeah.
And then the anti-inflammatory therapy, I think, is really kind of a paradigm shift. It's going to be hard to sell it to clinicians and to the pharma companies because many pharma companies are harking on type 1 interferon and producing, STING agonists and, CPGs, etc., and developing drugs such as simulation inhibitors that are inducing a type 1 interferon response. And of course, as typical of the pharma companies, they want to dose these drugs for long periods of time so they can make money, right? But, with a complete lack of recognition of how the immune system works. The immune system was not designed to withstand or tolerate long-term type 1 interferon signaling. The immune system evolved to sort of respond to rapid spikes and falls of type 1 interferon, which then unleash, innate immune cells such as dendritic cell activation and maturation, and then T cell activation, induction of an, NK cell activation, for example, which occurs through type 1. And then the system has to die down. Once you unleash those cells, you cannot chronically activate type 1 interferon, because we know now that patients and people that have much more high-level chronic type 1 interferon responses to viral infections, such as COVID-19, actually fare far worse and cannot control the virus, versus patients or people that actually have this really spike goes down. They don't have an exaggerated type 1 interferon response. And I think it's going to be really exciting in the coming years to figure out why there's these different populations of people, why some people get hugely sick, immunopathology, immunosuppression can't control the virus, while some people can. And I think we'll be seeing germline polymorphisms and differences in immunologic genes that regulate type 1 interferons, and even epigenetic changes and differences in people that have a more exaggerated long-term over-exuberant type 1 interferon response that causes immunosuppression and immunopathology, versus people that have a more attenuated response to interferon and in a short-term burst that is really what is the optimal scenario. So I think there's going to be some really exciting research, and I think that has bearing on cancer, because type 1 interferons and chronic and over-exuberant type 1 interferon and other innate immune signaling inflammatory mediators actually cause immune suppression through T cell exhaustion, induction of myeloid-derived suppressor cells, etc. So I think one of the hypotheses coming out of this is that there might be some epigenetic and germline hardwiring of how type 1 interferon responses are modulated, and that in cancer patients that have an over-exaggerated, too much of a type 1, too long of a type 1 interferon, are paradoxically the ones that do more poorly on immunotherapy and checkpoint blockade. And I think that's a biomarker we should be looking at.
I hate to interrupt this wonderful conversation, but
yeah,
we
sorry
are
about that. Yeah.
No, it's all wonderful. Thank you so much again, Laszlo. It was just wonderful.
Yeah,
and the amount of
now. Yeah, so I'll
yes,
head on over there.
I was just going to say the amount of interest really attests to the fact that you're always welcome in Guelph. Hopefully next time, August 8th, you said, in person, yes. And hopefully we will have the opportunity to interact with you face to face. Last but not least, I also wanted to again thank Dr. Clifford Chappel and Mrs. Chappel for this wonderful gift to us to bring some of the most renowned scientists in their respective fields to Guelph and to OVC, and really benefiting from their presence. So thank you again. To our thanks to the Chappel family. And also I wanted to thank Carrie Ann McGugan and Adrian Arnold, who did most of the work in the background behind the scenes, and also our communications team. So having said that, thank you so much again, everyone, and thank you again, Dr. Radwani. You have a meeting coming up. Actually, it's already come up.
Yeah, I'm going to be heading on over there.
Fantastic. So thank you again.
I'll move my electrons over there to the next meeting.
Fantastic. Excellent.
Okay. Thank you very much.
Have a wonderful afternoon.
Okay. Thank you. Bye-bye.
Bye now.